So, once again today, I think we've left the best to last, as I'm sure many of you will agree, having had me on first thing. So we are now going to hear from the medical director from Nuvalent, Dr. Viola. Yeah, Viola, is that okay? Yeah. Right. So with no further ado, I'm going to hand over to you. And because I want to hear from you, not me.
Thank you, Deborah. And I'd like to thank Deborah and the organizers for inviting me here to spend some time with you all. Last summer, I took my daughter to London for the very first time. Just two girls spending time together. I also have a younger son, so we decided to leave boys at home. It turned out to be the best family vacation ever. When we went home is Boston for now. We moved around a lot. So my daughter had a revelation. She came to me and said, "Mom, Boston is just a crappy version of London." So she's super, jealous. I'm back in London. And I promised her, after this conference, I need to do some shopping for her, so she'll let me go on with another business trip. So anyhow, I also feel it's extremely difficult to do a talk after Deborah's inspirational speech and Professor Ben Solomon's wonderful presentation about the history of ALK, but I'll try to do my best. So Nuvalent, and they forgot to put my name there, but that's okay. My name is Viola Zhu. So in my previous life, I was a thoracic oncologist and a phase 1 investigator at a cancer center in California for many, many years. Actually, my research field was targeted therapies, including, ALK, ROS1, EGFR. So, I was friends with, Professor Solomon, Professor Pape, but, I was kind of junior there, so looking up to them. And about 3-4 years ago, Nuvalent, approached our cancer center and then wanted to start a trial. Showed preclinical data to my mentor and me, and I was really just blown away. I was like, wow, this is gonna be the next upcoming drug in this field. And this was COVID, and COVID made people do a lot of crazy things. And I was really I was an associate professor. I didn't know what to do with my life at the time. And I thought, wow, maybe I can switch my career and join Nuvalent and develop this drug for patients. So that's where I am now. So this is about 3.5 years later. I'm still with Nuvalent. At the beginning, I led the phase 1 portion of our ROS1 drug, NVL-520, which now has a name called zidesamtinib. And I know this organization has some ROS1 patients here as well. So this trial is doing extremely well. It's at phase 2. And then I handed this trial to my colleague later on, and then started the phase 1 portion of NVL-655. And we dosed the very first patient in June 2022, a year after I joined the company. And then we finished the phase 1 portion in February of this year, so 20 months. 20 months, we dosed a total of 133 patients for phase 1. We transitioned the study to phase 2, and now the phase 2 enrollment is ongoing, is extremely active. We actually hope to finish the phase 2 portion of the study very soon. And then our plan is to present the data package to the FDA to get approval. So that's our inspiration. And I'll talk about some other plans with the company. So anyhow, with all this really, really long introduction, and let's talk. So my lawyer, our company's lawyer, made sure I mentioned this is our company's forward-looking statements. So anyhow, let's first talk a little bit about drug development. So I think some of you asked about this. This diagram really gives you a very simplistic view about the timeline of drug discovery and development. as you know, it's really a very, very lengthy process. I can tell you now, like drug development is very, very hard. So you may start with about 10,000 compounds here at the very beginning, okay? Let's see if the laser works. Oh, maybe not, but anyhow. So if you're lucky, you can advance about 250 into the preclinical stage, which is usually sort of the toxicology studies, on animals, right? And then just these 2 steps can take about 6 to 7 years. And then afterwards, maybe 5 lucky compounds by different companies can go into various phases of clinical trials. So we're talking about phase 1, phase 2, phase 3, right? And on the next slide, I will explain to you what different phases mean. So the Nuvalent trial, as I mentioned, finished the phase 1 portion, is at phase 2. We're hoping to achieve an FDA approval from the phase 2 trial. However, for the frontline patient, for newly diagnosed ALK positive patients, the current requirement by the FDA is we have to run a phase 3 study. So that's where we are right now as well. The clinical trial itself can take about 7 years or so. And then if you're really the lucky compound here, you may be able to advance to the FDA review stage. The review stage, by the way, can take about 1 to 2 years. So you can imagine there's a little bit vacancy here, because during that time, what do we do with the drug, right? This is usually an expanded access program will come in to allow patients to get access before the drug gets officially approved. So from the beginning to the end, it's about 10 to 20 years. It's very hard, really. Many, many drugs fail. Cambridge, Massachusetts is where our company is based. Well, it's actually the fake Cambridge, right? So anyway, it's a virtual company, so we all work from home. There's so many biotechs, but many companies actually fail and drugs flame out, really. So we have been really, really fortunate because our company was originally founded by some of the, I would say, the most brilliant biologists and chemists. They handed us some magical drugs to, for the clinical team to work with. So we have been very blessed. Part of the reason I'm still with Nuvalent right now. And by the way, I still see patients on the side, but it's only like half a day a week now. So I can't leave my practice. And I'm very grateful Nuvalent still allows me to do both. So anyway, so let's talk about different phases of drug development. Now, we don't really hear the terminology phase 0 often. This is usually the non-clinical pharmacology and toxicology studies. Now, phase 2. Phase 2, usually they are small studies to assess safety and determine the appropriate dose. We call the recommended phase 2 dose, RP2D. But phase 1 study used to be very small for a rare disease entity such as ALK. You could maybe run a study with 50 patients or so. This has changed. The FDA regulation has changed now. So think about our study. It took us to get 133 patients actually to advance to phase 2, which to me is a lot of patients, a lot of ALK patients, right? It's still a very small, very rare disease entity. Now, phase 2 studies are usually, again, based on this FDA definition, are conducted on small number of patients with the targeted disease to assess safety, dosing, and short-term side effects. I can share with you that we press released about 2 weeks ago. We already dosed 229 patients for the phase 2 portion of our study. Is it a small study? No, by the time we finish, this will be several hundreds. So again, I think that in the new FDA sort of regulatory environment, this is also changing. And phase 3, I would say for sure, these are usually studies in larger patient populations and with pre-established efficacy. Is it really for more varied patient populations? Again, I sort of disagree with this definition. As you know, we are going to launch our phase 3 study actually comparing NVL-655 against alectinib in the frontline setting. So we are actually looking at looking at a sort of a narrow patient population, this will be newly diagnosed treatment-naive ALK positive non-small cell lung cancer. Let's now talk, I just want to give you a very brief introduction on how oncology studies are assessing anti-tumor activity. The RECIST 1.1 criteria are most commonly used, I shall say. It's actually pretty kind of simplistic, I shall say. Maybe that's not the right word, but it's sort of the best we have, and I still think there's room for improvement. Essentially, every patient that enters into a clinical trial will get a baseline set of scans, including CT of the chest, abdomen, pelvis, and brain MRI for our study, okay? And then you mark all the tumors sort of based on this guideline. You can select up to 2 tumors per organ and up to 5 organs. So usually you go with the biggest. It could be 2 lesions, 2 tumors in the liver, 2 lymph nodes, for example, up to 2 lesions in the brain, or 2 lesions in the lung. Then you add them up, get a sum. Okay, this will be your baseline. And then every time a patient gets a repeat set of scans, this gets measured again. Then you get the new sum and compare that to the baseline. All right? So if there's complete disappearance of all target and non-target lesion, so the target lesion means any lesion that is about 10 millimeter or bigger, except for the lymph node. The cutoff for lymph node is about 15. So there's some caveats here. And all the other lesions are generally non-target lesions. So complete response, or CR, is disappearance of all lesions. Okay? Partial response is 30% or higher decrease in the size of target lesions, with some caveats here. The last one, progressive disease, or PD, is 20% or higher increase in the size of target lesions. And between partial response, PR, and progressive disease, PD, is stable disease, SD. And when we talk about overall response rate, ORR, that is complete response, CR, plus partial response, PR, okay? Sometimes you may see this terminology called clinical benefit rate. It's not often used, CBR, that is complete response, CR, plus partial response, PR, plus stable disease, okay, SD. So Deborah already shared this slide with you. So again, think about what I just mentioned. The red bars, they're all above the plus 20% line. So this is progressive disease, PD. The green bars are minus 100%. So this is complete response, CR. The black bars, this is below the minus 30. So this is partial response, PR. And the blue bars in the middle, this is stable disease, SD, okay? So with that, I'm gonna share with you the data we released very recently and presented at ESMO about 2 weeks ago. This is our comprehensive phase 1 data set. The phase 2 trial is actively ongoing, so we have not released any data yet. So. And I just want to point out that we are extremely grateful for the patients in UK that participated in the phase 1 portion of the study. I know Professor Anna Mitchum from Royal Marsden, dosed actually quite a lot of patients, during the phase 1 portion of our study. So I think Professor Solomon already shared this slide with you. Just very, very briefly, NVL-655 is a rationally designed ALK selective track sparing next generation or perhaps 4th generation ALK TKI. On the left-hand side, this gets really very scientific, but left-hand side, this is drugs activity against the various ALK fusions and resistance mutations. In the middle, this is showing you the drug's brain penetrance, similar to lorlatinib. On the right-hand side, I would say this is unique about this drug, is the avoidance of track inhibition. We want to avoid TRK because we want to avoid TRK-related neurotoxicities you may see with other ALK TKIs, particularly lorlatinib. And we were also very proud, to publish our preclinical data along with some case studies on cancer discovery. This was released, I think, simultaneously with the ESMO presentation. Let's see. I mentioned that we enrolled the very first patient in June 2022, and we transitioned the study to phase 2 in February of this year. So 20 months, 133 patients. This is really incredible when you think about it. In fact, our company's timeline, kept shifting to the left side, and everything was getting accelerated. This truly speaks to the enthusiasm and support of the ALK community. Okay, and we enroll for phase 1 advanced solid tumors harboring a ALK fusion or activating mutation. For patients with non-small cell lung cancer, they have to see at least one prior second generation or third generation ALK TKI. At the time, FDA essentially told us you could not dose any patient that only received crizotinib because this is a phase 1 study. You don't really have any proven efficacy, and patients can go on with all the other ALK TKIs. Therefore, this was not allowed. Similarly, we were not allowed to dose any treatment-naive patients because of the regulation, I think rightfully so, right? We excluded patients with concomitant oncogenic drivers because if your tumor has different mutations or discovered at the same time, targeting one mutation may not be the right way to do it, and that was the scientific rationale. So we dosed 133 patients, and 103 patients are non-small cell lung cancer patients that are response evaluable. We examined all 6 planned dose levels, okay, and then eventually selected dose level 5, 150 milligram daily as recommended phase 2 dose. So this is a daily dosing, and I think it is convenient for the patient. So as you can see, this is the patient. This table is showing you patient characteristic and treatment history. So this is really a heavily pretreated patient population. For example, about 56% of the patients, just look at all the sort of salmon color highlighted numbers here. 56% of the patients have history of brain metastases. 26% with compound ALK resistance mutations. That is usually after sequential ALK TKI therapy, such as crizotinib followed by alectinib or brigatinib followed by lorlatinib. The median prior line of anti-cancer treatment is 3. 84% of the patients received prior lorlatinib. So there's really no approved ALK TKI therapy in this space. And 44% received 3 and more prior ALK TKI, including a second generation and lorlatinib. Okay, I think Professor Solomon also shared this slide with you, but just remember what I mentioned about the waterfall plot. So I hope you can have a sort of a different perspective when looking at this data. So first of all, for the overall response rate for all dose levels, this is about 38%, okay, right there, left upper corner. And responses are higher among patients with baseline ALK resistance mutations. We then separated responses by patients with prior lorlatinib, 35%, without prior lorlatinib, 53%. Looking at the waterfall plots here, you can appreciate some of the deep radiographic responses for patients treated with or without lorlatinib. They are color-coded by the type of prior ALK TKI therapy. Full dots denote patients with baseline ALK resistance mutations. Half dots, single sort of excuse me, full dots a compound resistance mutation, half dots a single resistance mutation. Now, internally, I often, remind our teams that we are very familiar with numbers, with tables, with figures, but we need to remember every single bar represents a patient that entered our study, right? Gave his or her face to us, and then some of them actually derived a good benefit from this. So I think this is something we need to remember and not just bury ourselves, with all the numbers and figures and tables here. So we obviously care about responses, but we also care about how durable such responses are, meaning we want patients to be able to stay on the trial, stay on our drug or any anti-cancer therapy for as long as it can, right? So here you're looking at duration of response. Median duration of response for our phase 1 data set for all dose levels is 14.4 months, okay? The rate of duration of response of 6 months or longer is 78%. Again, we separated our data by patients with prior lorlatinib versus without prior lorlatinib. Okay. Here, you're actually looking at a subset of patients with baseline ALK resistance mutations. Median duration of response is 14.4 months. The rate of duration of response of 6 months or longer is 85%. Again, we separated this data sets by patients with prior lorlatinib and without prior lorlatinib. And some of these, subgroups still have very small number of patients, right? This is still a phase 1 data set, and we continue to follow these patients and, to hopefully get a more mature readout eventually. So I also think Professor Solomon, shared this slide with you. We care about CNS activity or brain activity, for many, many reasons. We want a drug to be able to get into the brain, to be able to treat tumors in the brain, to be able to prevent tumors from ever growing in the brain again, right? So here, looking at waterfall plot on the right lower corner, you can see the CNS tumor shrinkage for patients with measurable CNS lesions. It's a small data set, but we were able to see 1 out of 2 patients that are lorlatinib naive to achieve an intracranial overall response rate, okay? And then for the prior lorlatinib, the intracranial ORR is 2 out of 13. Right lower corner, this is called a swimmer plot. It's essentially showing you the duration of treatment, all confirmed CNS responders. So the green dots are for patients with CNS complete response. This is disappearance of all the lesions, target, non-target in the brain, okay? The pink dots are for patients with CNS partial responses. Very importantly, no CNS progression was observed among all confirmed CNS responders. At this data cut, the treatment duration was up to 14.4 months. Here, we're looking at one patient treated at a site in France. This patient previously received 3 prior lines of therapy, including chemotherapy, alectinib and lorlatinib, actually had CNS progression or brain progression on lorlatinib, entered our study, received a 150 milligram daily dosing, and was able to achieve a complete resolution of multiple baseline CNS lesions. The MRI imagings here are showing you one representative lesion that disappeared with NVL-655. The treatment continues at 11.4 months with ongoing confirmed CNS CR at the data cutoff. We previously shared 2 other case studies at the triple meeting last year. One patient had a confirmed CNS CR. The other patient had a confirmed CNS PR. Both patients are still, their treatment are still ongoing as of right now. Actually, as of the data cut. So now switching gears, let's talk about toxicity or safety profile. It is favorable and consistent with ALK selective track sparing design. On the right-hand side, you're looking at treatment-related adverse events or toxicities occurring in 10 or more % of patients. So what are these? ALT increased, AST increased. These are lab abnormalities, okay? Constipation, dysgeusia, which is altered taste, and nausea, okay? The rate of dose reduction is 15%, and the rate of discontinuation is 2%. Very importantly, the overall profile is consistent with avoiding track-related neurotoxicities you may see with other ALK TKIs such as lorlatinib. So I think Professor Solomon also shared this slide with you. As I mentioned earlier, the study is actively ongoing. The phase 2 portion of the study is enrolling extremely well. And it's a global study. We're right now enrolling both TKI naive patients and TKI pre-treated patients. Again, our inspiration really is to try to finish the enrollment and have a comprehensive data set ready to discuss with the FDA, hoping for an approval soon. With that, this study, the goal for this study is to get an FDA approval first for the previously treated patient population. For the treatment-naive population, we are launching a randomized phase 3 study globally. This is comparing NVL-655 against alectinib, because crizotinib, any drug against crizotinib cannot be done anymore. It's not really ethical anymore, right? So we spoke with the regulator. We landed on using alectinib as our comparator arm. We're going to launch this, really big global study, and then actually, I think we approached many sites in UK for this study, including several speakers at this conference. So we listed all the cancer centers that are currently participating in our study, but I just want to highlight that the current phase 2 study, phase 1, phase 2 study, this is not about Nuvalent, really. This is not even about the study investigators or site staff. This is really about all of you. It's about the ALK positive patients and their caregivers. And we're really, really grateful for your enthusiasm and support. Last but not least, I asked our internal team to mark our trial centers on this global map, and when I looked at this slide, I think I couldn't help but sort of feel humbled. Why? Because, I thought I was leading a global study, right? We have sites everywhere, right? But when you look at this map, really, there's still a lot of places, where patients don't have access to our trial. And forget about our trial. Patients don't have access to a lot of the existing ALK TKIs, in fact, to advanced technologies, right? So I can share with you because I was born and raised in Shanghai, and then I actually I'm a first-generation immigrant. That's always how I introduce myself to my American colleagues. Actually, I can't call American because it's not right for South America. I need to say United States colleagues. So I'm trying to learn all this culture, sensitivity stuff. So I did some of my medical training in Shanghai, in China. I sort of know a little bit about how healthcare is delivered in China. There's a huge difference comparing the two countries, really. So I really feel patients in the States are just so fortunate. because they have access. They have access to the best drugs. They have access to the most advanced technologies, and they have access to clinical trials like this, right? But in the end, now I'm not a politician. I'm here to speak on behalf of Nuvalent. But in the end, I think the global health delivery is the key here, right? We want everybody in the world, all the ALK positive patients, to have access, to be treated, so no matter where they are, because in the end, all lives matter. So with that, I'm going to conclude my talk, and I thank you for listening, and please feel free to answer any questions.
Unfortunately, you don't get off that lightly.
Okay.
And so I'm now going to open the floor out. Does anybody have any questions? Right, okay. Duncan?
Thank you. That was a really, really interesting presentation and really quite inspirational as well. So it's really exciting, the work that you're doing. Please carry on. So the question I had, and you touched at the start on these different measurements that the trials use for measuring how effective the drugs are. And sort of the headline one often uses ORR, which is obviously that complete response and partial response. But then you touched on the name of the other one, which I think was CBR, clinical benefit rate. Now, I understand why ORR is the one that the oncologists pay a lot of attention to, but as patients in the support groups, there's a phrase that gets used an awful lot, and that phrase is stable is good. So we saw an ORR rate there, but I don't know if you have that CBR rate as well, and what sort of figure that is, and if that's significantly better than the ORR.
That's a very good question. We internally do have that data. We have not released. I completely agree with you. In clinic, if I see stable disease, I'm actually quite happy, right? Particularly if somebody has been treated previously with multiple ALK TKI therapies. And traditionally, for less heavily pretreated patients, the thinking is if you have a strong inhibitor, you're going to hit the target very hard. Therefore, you should be able to see a better response, like CR or PR. So the ORR is sort of a more rigorous way of evaluating a drug's efficacy. For some drugs that don't sometimes it's not about the drug, it's about the target. For a target that cannot be blocked very effectively, some companies then decided to add a stable disease, to sort of, boost the number a little bit for presentation, I'll tell you. So we're happy with the ORR data, but obviously, when we give the whole data set to the FDA, FDA is going to look at the stable disease as well. And then FDA also wants to look at is the duration of response, not just ORR, is how durable the response is, which is actually a key study endpoint for any phase 2 study right now. So,
Okay.
Thank you.
Mm-hmm.
Any other questions?
Oh,
Yes.
yeah. At the moment, we're on alectinib, and we've been offered loritinib. And if we were to go onto the Nuvalent version, how would we get onto a trial of that in London?
Okay, so I wish Dr. Pape was here. So I think we have 3 sites in the UK that are actively enrolling patients. One is Royal Marsden. One is in Scotland, and then the third one I'm blocked. Manchester, right? Oh, thank you, Deborah. So essentially, what I would suggest is one way is to first talk to your treating oncologist, and then have a discussion first, and see if that's the right way. Because if you're doing well on alectinib, there may not be a reason for you to switch your therapy at all, right? Now, if at any point, your oncologist wants to switch, or you actually do not want to receive alectinib, want to switch, then you can have your oncologist make a referral to one of the 3 trial sites, and then all cohorts are open. Then after the first wave of assessment, you may be able to get on the study. One thing about our study is we do give travel reimbursement. We have a cap, but if you're traveling within the area, for the trial, it's actually being covered. So.
Any other questions? Oh yes, over there.
Thank you. I have a question about the phase 3. You said that only treatment-naive ALK positive patients will be included in this study. So how does it affect people who have been on TKIs before? So what does it mean for the rest of people? So they were included, obviously, in phase 1 and phase 2. So when it comes to the FDA approvals and other approvals, how would we access this TKI?
Okay.
Would we be able to get the approval somehow?
Yeah, so I can comment as much as I sort of know in terms, because I don't know much about how NICE sort of functions. As I think this question was asked, there's usually a bit of a delay, from the time FDA approves a drug to how this drug is approved and reimbursed in UK. At this moment, the phase 2 portion of the study is actively ongoing. Actually, every single cohort is open. I didn't mention this. The cohort design is based on the prior anti-cancer therapy, and actually every single ALK positive patient can potentially get on the phase 2 study, because we have treatment naive. We have one cohort for treatment naive patient, one cohort for only 1 prior ALK TKI, or 3 or more ALK TKI, et cetera, et cetera. Now, our phase 3 study is to be launched in Q1 of next year. So that study is only for treatment naive patient, by the way, as you mentioned. We will keep our phase 2 study open for as long as it can until we hit the point where we have enough data to get FDA approval, while the phase 3 study is actively ongoing. During this gap, when we have enough patients for the phase 2 study to the drug approval, as I mentioned, there's a FDA review process that's about a year or a year and a half or 2 years. We're going to launch sort of expanded access program in order to allow patients to get access to the drug. How that program is going to play out, we don't know yet. We're just at the very beginning of thinking about this, thinking about where we want to go for the expanded access. So does that kind of answer some part of your
maybe
questions?
I rephrase it. I would just say that would the phase 2 be sufficient? Would it generate enough data for you to apply for the FDA, or you will still need the phase 3 for people who were not treatment naive?
I think you asked questions like some of the regulators in our company, because we get asked a lot about our plan, clinical development plan. So our goal, so first of all, the drug has received FDA breakthrough therapy designation. By the way, I forgot to mention this. This is a sort of a recognition of a drug in the space, and then it will lead to a sort of a faster approval timeline. So our breakthrough therapy designation is for 2 prior ALK TKI, so the 3rd line setting. Now, because the data so far have been really good, at least how we feel, we hope with enough patients, with about, phase 1 has 133 patients already. Phase 2, we already dosed 229 patients. And by the time we finish the study, I think it's going to be, 400 or 500 patients. We hope to use that data set to really break down each cohort and to get an FDA approval for any previously treated patient, not just the 3rd line, even for 2nd line. That's our goal. And once we get that approval, then the only space that doesn't have the drug's approval is the front line, in which we will that's why we need to do the phase 3 study. That's sort of the thinking behind the clinical development of this drug.
Okay, thank you. And just out of curiosity, people who were enrolled on phase 1 and now phase 2, would they be continuing to be on this medication after they finished the trial time?
Yeah, that's a very good question. I think that there's sort of an obligation for us to provide the drug for as long as the trial is ongoing, right? Even, but once the trial is completely closed, there needs to be a mechanism to either allow them to stay on drugs or transition them. If the drug is approved, it's easy. For countries like UK, if there's a delay in the approval process, it's something we're discussing internally. I cannot promise you anything because I don't make that decision, but we obviously have to map out the region. It's very complicated. Each country has different process. We need to think about the access, but that is something Nuvalent is working on right now. I just can't give you a definitive answer yet. Mm-hmm.
And I cannot resist to ask this question. Do you have any new TKIs in the pipeline for ALK?
Our investors asked us this question a lot to sort of value to think about the value of the company. I'm not at liberty to disclose anything except that we have the ROS1 trial at phase 2 portion at the phase 2 portion of the study. We're hoping to actually get approval for we call line agnostic, meaning we're hoping to get approval in any space for the ROS1 patient, because ROS1 is even a smaller space. So I don't think there's a need to do a randomized phase 3 study to get approval for the frontline setting. I talked about ALK. We have a HER2 exon 20 TKI. The phase 1 portion has just kicked off. It's currently just in the US. As soon as we have some preliminary data, again, HER2 exon 20 is a small target for non-small cell lung cancer patients as well. I think the percentage is about 2% or so. It's kind of like ROS1, a little lower than ALK. The idea is once we get to a good dose level, because you have to start at a small dose and go up. Once we get to a good dose level, we're going to do a dose expansion. Then we will have that study, more globally. So, and then beyond that, our company, was really originally founded by biologists and chemists. So they're actively working on a lot of other targets. One thing we often do is, we meet with all the experts at conferences and then we pick their brain. In fact, I'm gonna meet with Professor Poppe after the conference. One thing to discuss is in the clinic, where is the unmet need? What about MET? What about RET? What about AKT? What about all this? Do you see a clinical unmet need? We constantly gather that feedback and bring it internally to think about what's next. But as you know, I feel really humbled, just for being in industry for about 3.5 years. It's very tough. So we internally don't pressure our biologists, by saying, hey, you have to advance one drug to the clinic every year. Some companies do that, so biotech is actually ruthless. So some companies have this very, very strict timeline, but we don't, because we want them to be able to, do the best, they can, and when they feel ready, when they feel a drug meets all these characteristics, they're ready to advance to the clinical stage, then we take over. So that's where we are. Mm-hmm.
Yeah, Professor Popat mentioned that a lot of TKIs for ALK are coming out of China, and they already passed their clinical trials. So is it something you may look into with your company?
Yeah, I know. I think there was an ALK TKI actually presented at WCLC in September also. It was the 9th ALK TKI in China. So Yes, that's another country where we don't really have presence. That's something we're working on. Definitely, there's there are a lot of talented, people there as well. It's a competitive space, so something we're sort of monitoring and then see, where it goes. Yeah.
Can I
Thank you.
ask everyone to give their final thanks to Viola? Because I think we've all found it fascinating and incredibly inspiring as well. So thank you very much indeed.